Namaste students. I am Jayoti from the Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth University, Jaipur. Today I am going to discuss about the topic control of microbial growth by effect of heat. Physical method of Physical method of the control of the microbial growth. So what are the different physical methods? First is the heat. Heat kills most of the microorganisms by denaturing their enzymes and their other proteins. Resistance varies widely among the microorganisms. Different type of heat can be applied to inhibit and to kill the microorganisms. So some terms which need to be understood before going to the heat killing of the microorganisms are that thermal death point, thermal death time, and decimal reduction time. What are these terms? And what is the meaning of these terms? Thermal death point is the lowest temperature at which all of the microbes in a liquid suspension will be killed. It is the lowest temperature at which all the microorganisms will be killed in 10 minutes. Thermal death time. Okay, what is the thermal death time? Death time. Thermal death time is the minimum length of time in which all the microorganisms will be killed at a given temperature. For example, suppose that we are killing all the microorganisms at the 80 degrees centigrade. Okay, we are going to kill 80 degrees centigrade, kill all the microorganisms at 80 degrees centigrade. So what will be the thermal death time? For example, one microorganism has been killed at 20 minutes, one microorganism has been killed at 30 minutes. Okay, one microorganism has been killed at 40 minutes. Different microorganisms will have different thermal death time. Okay, but the temperature is same here. The time is varying. Right? In the thermal death point, we are giving same time. We are giving same time, but the temperature is varying. For example, we are giving only 10 minutes and we are increasing the temperature. For example, we are increasing the temperature from 60 degrees centigrade, 70 degrees centigrade, 80 degrees centigrade, 90 degrees centigrade. So that is the thermal death point. Now, decimal reduction time. Decimal reduction time is the time in minutes at which 90% of the bacteria at a given temperature will be killed. Okay, so 90% of the bacteria will be killed in a given time. Okay, at a given temperature. Okay, and mostly decimal reduction time is used in the canning industry. Okay, sorry. What is the most heat which can control the growth of the microorganisms? So different heat are used: moist heat, dry heat, and uh, heat of the steam. Okay, so we are going to discuss moist heat. Moist heat kills microorganisms by coagulating their protein. The proteins will be coagulated and it will kill all the microbes. In general, moist heat is much more effective than the dry heat. Okay, it is very effective because it will kill all the microorganisms of any object. For example, some objects need cannot be treated with the dry heat. Okay. For example, the plastic waste cannot be heated in the dry heat. So we can we can use the most heat. Okay. Next is the boiling. Heating to 100 degrees centigrade or more at sea level kills vegetative forms of the bacterial pathogens. And almost all viruses and the fungi and the spores within 10 minutes or less can be killed. Endospore and some viruses are not destroyed this quickly, and some brief boiling will kill most of the pathogens. Like hepatitis virus can survive up to 30 minutes of the boiling, and endospore can survive to 20 hours of the boiling. So these microorganisms need more time to be killed. Some reliable sterilization with most heat requires temperature above that of the boiling. So the one important techniques in the most heat is the autoclave. Okay. Chamber which is filled with hot steam water, yes, sir, and it is a preferred method for the sterilization unless material is damaged by the heat, moisture, or high pressure. In autoclaving, the temperature will reach up to 120 degrees centigrade by the high, high steam pressure. The pressure allows the temperature to reach at 121 degrees centigrade. Because if we have temperature and if we have pressure control, then the temperature can be reached at 120 degrees centigrade. And at the 121 degrees centigrade, most of the microorganisms and the spores will be killed. And all organisms and those spores are killed within 15 minutes. And it requires more time to reach center of the solid or large volumes of the liquid. Pasteurization is also a technique for the most heat killing. Pasteurization is a technique which is developed by the loose pasture to prevent the spoilage of the beverages. It is used to reduce microbial responsible for the spoilage of the beer, milk, wine, juices. Okay, so it is very important technique and developed by the loose pasture. And classic method of use of pasteurization is that milk is exposed to 65 degrees centigrade for 30 minutes. That is the classic method of pasteurization. High temperature short time pasteurization. Okay, it is used in recent uh, time, and milk is exposed to 72 degrees centigrade for 15 seconds, and then immediately transfer to the cold environment. And the high temperature pasteurization UHT. In this technique, the milk is treated at 147 degree for 3 seconds, and then it is cooled very quickly in a vacuum chamber. Okay. So these are the three techniques: classic, high temperature sorting, and ultra high temperature pasteurization. The advantage is that milk can be stored at room temperature for several months using this pasteurization technique. Okay. Now, what different another method of the heat transfer? Heat transfer for the killing of the microorganisms. First was moist heat. Now the second one, that is the dry heat. What is the use of the dry heat to killing the microorganisms? The dry heat kills the microorganisms by the oxidation effects, and it kills the microorganisms by direct flaming. Okay. We can use some flame to kill the microorganisms, like we do it for the killing the microorganism on the surface of the inoculating wood and inoculating needles. So we can apply the direct flaming to the wood so that the microorganisms which are present in the wood will be killed. Heating mantle is used in this method. Uh, Next technique is the incineration. Incineration. This is also a dry heat method. Incineration. It is effective way to sterilize disposable items like paper, cups, dressings, and biological waste. Next technique, the hot air sterilization. We need to place objects in a oven. That is the hot air sterilization. We are using a hot air oven. Okay, and in hot air oven, it requires 2 hours at once, 100 to 170 degrees centigrade for the sterilization. And dry heat is transfer. Dry heat transfer less effectively to cool body than moist heat. So it transfer the heat very effectively than the moist heat. But it has some disadvantage that all the objects cannot be uh, treated by the dry heat. Okay. So these are the techniques which are used for the uh, heating of different objects. Some more techniques of the sterilization will be discussed in next video, which are also the part of the physical method of the microbial control. So my lecture is over here. If you have any query, you can ask me. The question which can arise from this lecture is that what are the different techniques for the killing of the microorganism using heat. Okay. So students will be better able to answer all these questions. So my lecture is over here. Thank you very much.